Friday. It's Friday, right? I'd say it's almost the best day of the week. Yeah. A little bit. All right. Any questions over the material so far? We have quiz that's due today. Yep, 11:30. Do you put the due date on there? Due date and time. So if you get onto Blackboard calendar, it should list like what's due. You can thank a graduate student for letting me know about that. Had no idea. So you can make sure you do that. We are moving on. All right. So gave some introduction about Hardy-Weinberg. Hardy-Weinberg equilibrium. We'll talk about that. We'll work with that equation. Then not next week, the week after that, because no lab next week. And that serves as a very good way to identify which populations are actually evolving. And if they're evolving, then we kind of have to go back and look to see, well, why? What's the process? What's that mechanism? Are they just evolving due to random chance, so genetic drift? Is there gene flow? Is it mutation that's popping up? Or is it some form of selection? Either differential reproduction, so probably would hear that as sexual selection, or is it a natural selection, differential survival? But as I said, differential survival and reproduction together is natural selection. That's what we're talking about. All right. So what we're going to do now is to talk about the origin of adaptations. How do they evolve? Not just like natural selection, but what's the general process? What do we need in order to get this? And you'll see that it's not necessarily just evolution, but we also have to take into account ecological processes as well as genetic processes inside of our population. All right, so recall Darwin's tenets, those 3 tenets. What's the first one? Ooh. All right, let's try this. Let's turn it back on, turn it back off, see if it works. And if not, I'll grab some new batteries. Still staticky? Those of you listening in on YouTube, the audio is staticky. So I take all of the yes as static. Shoot. Get that out. All right. All right. Let's swap batteries. Oh, hold on. Maybe that was it. Try that. It wasn't plugged in all the way. Still static. Still static. All right, fresh batteries. Let's try this one. Still static? Maybe I need to talk a little bit more to get the static. Still static. All right. All right, this one should work. A little bit different. This should clear up the static, hopefully. We can work to get this one fixed. Let me know if it's still static, because we're using a different microphone now. That's better. All right, so you guys hear it coming out of these speakers at all? All right, I'll put in that request. See what we can get. All right. First tenet. Someone said something. What was it? Okay, heritable variation. We need heritable variation. What's number 2? All right, limited resources, and what's our third? Yep, so if we have a trait that gives an individual an advantage to get those limiting resources, then they should leave more offspring. And that trait then spreads throughout our population. All right, so the whole thing is all about this heritable variation out there. All right, we need it. All right, if we don't have that variation, if all of our individuals are identical throughout the population, then there's nothing that selection can really act on. All right, if it acts, it's going to take everyone out, or everyone's going to do just as well as everyone else, and nothing happens. We stay stasis. All right, so what we need to have is some variation. We have to have some way in which individuals can be better at getting those limiting resources than other individuals. So there's 2 broad sources of this variation. All right, there's variation that's due to the genetics, and there's variation that's due to the environment. We refer to these terms as being nature versus nurture. All right, variation that's caused by nature is all about the genetics. What does your genome say you have? The nurture component is the environment. How much of an effect does the environment have on our traits? And our traits are going to be our phenotypic traits. When we have this environmental effect, we see phenotypic plasticity. All right, so what is phenotypic plasticity? I know I talked about it in my lab that first week. It's defined as the ability of an organism to produce different phenotypes in different environments. Give you time to write that. All right, phenotypic plasticity is the ability of an organism to produce different phenotypes in different environments. I just had my notice. I've got the attendance Blackboard quiz that went live at 8 or 8:05. Don't click it yet. I still have to give you answers. The phenotypic plasticity is the ability of an organism to produce different phenotypes in different environments. Our genome could be the same for 2 different individuals, but if they're raised in different environments and they have the ability to exhibit phenotypic plasticity, you're going to get different phenotypic traits. Now, you can say that selection can now act because they have this variation, but it actually can't act on that, since that variation isn't due to the gene itself, it's due to the environment. All right, so an example of this is in our diagram here. Stomata density on plants. If you raise the plant in a high CO2 environment, you don't need a whole lot of stomata for gas exchange, so you see very few or a low density of stomata on that plant. If you raise it in a low CO2 environment, now the plant's trying to get as much CO2 as it can, needs to maximize gas exchange, it increases the density of your stomata on our leaves. Did the genes, did our genetic code actually cause this difference? Not directly. It was the environment. The plant sensed what environment it's in and responded accordingly. Now I could step back and say, the ability to be phenotypically plastic, that is a trait on which selection can act. But these different traits produced from phenotypic plasticity really isn't. Yes, in the sense that adaptation isn't referring to the genes, to the genetic code. So this is where our terminology kind of gets mixed up. You kind of have to understand that selection could produce individuals that are phenotypically plastic. And we'll talk about situations when that's favored. Right? Or you can have selection favor those individuals that aren't phenotypically plastic. And then you've got that next step where phenotypic plasticity allows individuals to survive in various environments, but if they end up getting a trait like this one that says this is really good for the plant, selection can't really produce a bunch of plants with high stomata density because this isn't coded for in the genes. What was coded for is this break, this split to say we can go either way. Keep that in mind. All right, so we have our phenotypic plasticity. I'll draw another example because we'll talk about how we can distinguish between phenotypic plasticity and genetic variation. So how do we distinguish between the nature and the nurture part? Let's talk about the nature. The nature part is everything that's caused by genetic differences. Selection is going to act on this because our genes can be heritable. Not all of it is heritable. Not all of it is heritable. The ones that are heritable, we refer to it as additive genetic variation, something that can be heritable. There's also genetic variation like maternal components, methylation sites. Right, that's all caused by the genes, but you don't really inherit that. Or selection can't really act on that. All right, so what we can do is model how quickly selection acts on our population. Tri-beta meeting, 6 p.m. Wednesday, just a reminder. All right, so we have some equations that we can do, that we can use. All right, and I'm going to introduce some math just to kind of get to the main equation. All right, because what we want to know is how quickly will selection act, or how quickly will our population change? So let's first talk about our P's and Q's. Oops. Increase this. Let's move this up a little bit closer. All right, so if you've looked ahead to the lab, you've seen that lowercase p refers to our allele frequency. That's going to be our frequency in our population, our observed allele frequency. All right? P prime will be our predicted allele frequency in the next generation. All right, so what Hardy-Weinberg is going to be interested in is predicting our allele frequency. And we can predict it assuming that our population is in Hardy-Weinberg equilibrium, meaning that there's no evolution going on. When we incorporate selection, our new allele frequency will be the old frequency divided by 1 minus SQ squared. All right, so it's our old frequency, and what we're doing is basically removing individuals from our whole population because of selection. So if we go back to what S means, S is our selection coefficient. We introduced that in our last presentation. All right, this represents the proportion of our homozygous recessive individuals that don't make it to the next population or to the next generation. Our Q square is our predicted proportion in our next generation. So if Q squared is what we predict in our next generation, we have to remove a certain percentage of them, a certain proportion of them because of selection. All right, we'll do this in lab. We'll actually utilize this equation. All right, we'll utilize this equation. What I'm interested in is the difference. So the change in our allele frequency will be equal to our predicted frequency, this P prime minus P. Because this tells us how quickly our population will change. And that's what we're interested in. How quickly will selection act? Well, we already defined what P prime is. It's this equation. This equation right here. So what I'm going to do is substitute that. I'm going to substitute this in for P prime, simplify, and get this equation here. SPQ squared divided by 1 minus SQ squared. So once we substitute and we simplify, this is what we get. Do you have to memorize this? No, you don't. No, you don't. Do you have to memorize this one? No, you don't. We'll work with them in lab. All right, and just from practice, you'll probably start to somewhat memorize. But this equation kind of holds the summary of how selection acts on our population. All right, so the rate of change, how quickly our population will move due to selection, is going to be dependent on this value, our selection coefficient, and it's going to be affected by our P's and Q's. Now, P's and Q's are related. All right, P plus Q equals 1. We're dealing with proportions, two proportions. They have to sum to 1. So if P goes up, Q goes down. If P goes down, Q goes up. So they're related. All right? So when we look at this equation and we say, well, if we want to increase how quickly our population will change, what can we do? Well, if we increase our selection coefficient, if we increase this S, it causes this to go up. And if our numerator goes up, our rate of change goes up. That makes sense, right? So if we look at our graph on our slide, that's what it's showing. We have different values of our selection coefficient. As our selection coefficient increases, we approach that 1 much more quickly. So at our lowest level, at a selection coefficient of 0.1, we increase slowly, we pick it up, and then we slow down. And let's say this is 100 generations. It takes 100 generations to get here. If our selection coefficient increases to 0.5, now we get there in maybe 70 generations. If we increase to a selection coefficient of 1, so 100% lethal, 100% sterile, now we can get there in maybe, let's say, 40 or 50 generations. All right, our population changes very, very quickly. The second component of this is that P's and Q's. All right, now what we're trying to do with our equation is, again, maximize our numerator. Make this as large as possible. Well, how do we do that? Well, if P and Q are equal, then this term will be the highest that it can be. As these terms, as P and Q starts to deviate from 0.5, that value, that product will actually get smaller and smaller and smaller. So if we want to have the fastest rate of change in our population, we want P and Q to be about equal, which is right at 0.5. So I've marked on this figure the points in our population that are exhibiting the fastest rate of change. For those of you that have had calculus, maybe even pre-calculus, what is this point? What is this point on our graph? I bet a couple of you have had it. What's the point on that curve? Anybody? Let me ask. Waiting on anyone at home. Let's describe it another way. This spot, this curve at this point is curving up. So it's a concave up curve. This curve is curved down. That's a concave down curve. That transition point that has a name, what is that? Anybody? We're on a delay on YouTube, so I'll have to give it 10 seconds for any YouTube watchers. I guess no one had calculus. I just find that hard to believe. All right, that's our inflection point where we switch our concavity. Did you know that? Inflection point. What's that? Still covered it. It's our second derivative. It's 0. All right, so that's our inflection point where we switch, where we flip. That's our point where we see our fastest rate of change in our population. And it doesn't matter what S is. All right, no matter what, from 0.1 all the way up to 1, that's going to represent our point of fastest change. And we'll see this again. We'll see that inflection point again. All right, take a brief sidetrack just a little. Our quiz has 2 answers. That's just for a number. So 515 is going to be our first answer. You're going to have to keep watching for our second answer. All right, so don't start that quiz yet. All right, so these 2, selection coefficient and our P and Q, represent the first part of describing how quickly our population changes. All right, and that covers our equation. This is what we would cover. The third thing that affects our rate of change is heritability. This term describes how much of this variation is actually controlled by our genes. And hopefully it's kind of intuitive. The more variation that's controlled for by our genes, the faster selection can act. The less it's controlled by our genes, the harder it is for selection to act. All right, so heritability, H, is defined as the proportion of phenotypic variation for a trait that is determined by the additive effects of its genes. So this is again how much of that trait is controlled by the genetic code. We see this and we can measure it by quantifying similarity of offspring to their parents. So the more similar the offspring are to their parents, the higher the heritability. The larger the H, the more the trait is controlled by the genotypes. So if a trait has an H of 1, so it's going to range from 0 to 1, just like our selection coefficient. If our heritability is equal to 1, that means that our trait is 100% nature. The trait, the entire trait, is controlled by the genes. And that means that when we take individuals and mate them, their offspring will show a very high similarity. And it's going to be a consistent match to the parents. If our heritability is equal to 0, then that trait is determined 100% by the nurture component. It's determined entirely by the environment in which that individual was raised. So for us, we see this trait as appearing somewhat randomly. All right, we made our parents. It's going to be random what we see in our offspring. They may match the parents. They might not match the parents. Who knows? But the more our H increases, the faster selection can work on our trait. Right? So how do we include it in our equations? Do I not have this? I'm going to scroll ahead. Guess not. I need to. We don't work with the equation, so I don't see a reason to include it here. There is response to selection, and I believe will come up again with the response to selection. But know that, yeah, the larger the H is, the faster selection can act. So selection is affected by our selection coefficient, the value of our P's and Q's, and our heritability. Ready? All right, so this process of individuals being able to respond to the environment in which they're raised can lead to an interesting group, interesting phenomenon. And that is our variation, our environmental variation, could lead to the development of what's called an ecotype. Now, usually we see this from the from the geographic variation. So in St. Angelo, we can go to 2 or 3 different locations where we can find the same plant species, and those subpopulations in those areas may actually exhibit this ecotype characteristic. All right? The geographic variation is due to the differences in the environment, or I should say, the ecotype generation is due to the variation in the environment, and because the variation in the environment follows geographic locations, we recognize it as geographic variation. So what's an ecotype? An ecotype is defined as a genetically distinct population that is adapted to the local environment. Same species. We identify, we key it out as the same species. However, when we sequence them and start looking at genetic similarity, genetic differences, we can identify distinct groups that are based on geographic location. And when we look closer, we see that that variation, that geographic location differs from other geographic locations. All right? So the development of these ecotypes really is a consequence of 2 things. 1, the properties of fitness, and 2, this idea of heritability. All right, so the properties of fitness, quick review. What are the 3 properties of fitness? Say what? Property of fitness. Okay, so it's a property of the genotype. Same genotype, same fitness. Property of the environment. There's our ecotype. And we measure it over more than 1 generation. All right, so fitness is specific to the environment. You change the environment, you change the fitness. So that can generate new selective pressures leading to genetically distinct populations. And the second thing, in order for that to happen, selection must be able to work. And it needs that heritable variation. So the variation that we see, the phenotypic variation, must be controlled at least partially by the genes. It doesn't have to be 100%. And in fact, a lot of our quantitative traits aren't 100% controlled by our genes. All right? So the ecotype formation develops because of the properties of fitness and because of the heritability in these traits. Our example that we can use is serpentine and non-serpentine soils. Does anyone know what serpentine soils are or know their characteristics? And that's what this graph is. We've got serpentine and non-serpentine soils. And this is common yarrow, or yarrow. Anybody? The serpentine soils are pretty harsh soils. California, out in the mountains, Cascade Mountains, mountain ranges, they have patches of what's called serpentine soils, which is high in some of these heavy metals. And it's hard for plants to grow in it. All right, but some of these plants can grow. So here you have a forest, and we'll use this entire floor here and say this is our soil area, and we've got this plant. I'm not quite sure. I don't think these things. I think they can grow larger, but I think generally speaking, when they're going to be fairly small. So we throw out that yarrow, you let them grow. But I'm going to tell you, throughout here, we can set up the tiles in like a checkerboard pattern. So we've got these wider color tiles, and then we could paint them in and say black color. And what we're doing is setting up a geographical variation in our environment. So we'll say these wider ones are the serpentine soils that are very hard and harsh for the plants to live in, and then the darker colored tiles represent our good soil, say good soil, all right? Through selective processes, what we can then get is, maybe yarrow growing all across this area. And it looks like it's one homogeneous population. But when we go and sequence those plants, what we see is that those plants on the light colored tile, they're one genetic population. Those plants growing on the dark tiles are a second genetic population, and they are genetically distinct. Those are ecotypes. Well, why did they develop like that? It's the serpentine soils. It represented a very harsh selection pressure, and those plants had to figure out a way to deal with the heavy metals. They had to deal with the heavy metals. And they learned how to do it, but they needed the genetic variation to allow them to grow that way. And that's what this was. You can see serpentine, non-serpentine, serpentine, non-serpentine. You've got your plants, and some of them do well in serpentine, some of them, they don't do well in serpentine. And then when you switch it, so these are the non-serpentine soils, these are the serpentine soils. You can see some of them do well, some of them don't. Those are our ecotypes. All right. So we'll talk about these ecotypes, the development of these ecotypes a little bit later, because it pertains to also migration and overall selection pressures and so forth. But since we're talking about variation, physically or visually, they look identical. We don't see this ecotype. It's only when we start looking at the genes where we realize, yeah, these are genetically distinct. Questions? You ag majors, have you taken soils yet? Some of you have. You had soils? They talked about this, didn't they? They mentioned serpentine soil. Yep. Yeah, that's these serpentine soils also they tend to have poor water retention. So you see some of these adaptations for arid environments, some of those adaptations evolving. It's rather interesting. Now, I did the checkerboard pattern. It's a little bit bigger than that, so maybe we'd have to do like 4-foot squares. That's kind of how it looks out in nature. All right, moving on. We would think that selection can always produce the adaptation that is best fit for that environment, right? And that's what we talk about. That's probably what Bio 1 and Bio 2 were talking about. Selection's always going to increase your fitness. And that means then, or by extension, then you would think selection's always going to produce the best adaptation. But we know that when we look at adaptations, it's not. A lot of times we don't see that optimum solution. And it exists all throughout the animal and plant world. Non-optimum solutions exist. Not everything is going to be the best fit to the environment. Do they have to be? I'd argue if they're good enough just to get by, hey, that works. All right, but the question then becomes, why do they exist in the first place? Why have they persisted? Why didn't selection produce the best adaptation? Three explanations. Number one, our adaptations are contractions. Number two, evolution is a continuous process. And number three, we have to deal with constraints on evolution. So what we're going to do is hit each of these points and explain each of these 3 points as to why the optimum solutions are very rare out in nature. So reason number one for non-optimality, adaptations are contractions. When selection works, selection is going to act on traits that exist. It needs that variation to exist. And in some cases, selection is going to act on some trait, some current adaptation that might be slightly unrelated to what it needs, to the problem that it needs to solve. So selection then produces or generates a modification of what the organism already has. So an example is the panda's thumb. Famous example. All right, do a search for the panda's thumb. Can't remember if it's in Science or Nature magazine, but I believe we have access to it, so you can download the actual paper. Now, what is the panda's thumb? Do pandas have thumbs? Yes, because I used it up here, right? Panda's thumb? You say no. Why not? What is a thumb? Yeah. What bones are those? Some of you have also had anatomy, right? What's that? Phalanges. A panda has something that looks like that. Are those phalanges? Almost looks like it. And those are phalanges. They have something that looks like that. It looks like a thumb. Make a joke that if it looks like a duck and it walks like a duck, it must be a dog, right? It's kind of where we're going. It looks like a thumb. It actually kind of functions like a thumb. But it's actually not a thumb. It's a modification of the radial sesamoid bone, which is in the palm. All right, it's one of your metacarpals, I believe, in human anatomy. All right, so why? Well, we have to understand panda's diet. Panda feeds on what? What's it feed on? Bamboo. What does it do? Has anyone sat and watched a panda? They do. They peel the leaves off, right? I didn't get to see a panda do it. When I was in Memphis, they brought a panda to the zoo, and I thought, oh, that's going to be cool to go see it. They were charging $10 to get to that exhibit to go watch a panda. Like, hey, you know what? I'll watch them on YouTube. But yeah, they peel off the leaves. Is it a problem for us? No, crap. We've got a thumb. Does a panda have it? Actually, X out the panda and say, does a grizzly bear have it? No, a grizzly bear doesn't wave with their thumb, right? So, panda needs to peel these things off, and you can struggle. But just imagine if you have one mutation that caused that radial sesamoid bone to project out a little bit. Doesn't need to project out much, but just enough that gives that one individual a little bit advantage at removing those leaves. That means the panda spends less time stripping off the leaves from the bamboo shoot, more time eating, more energy, more offspring. All right, so then their offspring have that little nubbin on their bone. The longer that bone gets, the better they are at stripping off those leaves. Until eventually we get this, what looks to be a thumb. Very efficient, maybe not very efficient, but efficient enough to where the panda is able to rapidly and quickly remove off those leaves to acquire food. It doesn't have to be the optimum solution. It only had to be a solution that allowed that individual to have a higher fitness than others. And then selection starts to do the rest. So it's a contraption. It's just a modification of what we currently have. Questions on that? Point number 2. Oh, hold on. I think we're going to stop here. So stop at the panda's thumb. We need our second answer. So quiz asks what 2 numbers are the answer. 515 is the first one. Second one is 711. 711. 2 different numbers. Just one question. Multiple answer. Yep. Multiple answer type question. Make sure you pick both of those numbers. 3 minutes. Go ahead and do it. No class on Monday, Labor Day. Enjoy the 3-day weekend. All right, no lab next week. We'll come back here and meet on Wednesday. We are, I think, approaching. How many slides are we? This is slide 6. There's a total of 16 slides. I'm not sure. I don't think we'll get through this all on Wednesday. Get through the rest on Wednesday. So we'll probably finish this up on Friday, and then another quiz will go live on Fridays once we finish this up. All right, don't forget the quiz tonight. 11:30. 3 attempts. Highest grade. I'll keep the highest. All right. After you, yep.
My Blackboard won't let me on. It keeps shutting down.
No. Blackboard sound. No, we won't do that. See if I can get in. Oh, I can get in. Grades. It's open until 8:55 is how long grades open. See people taking it. But not the website? All right. Who didn't take it? All right. Here. Foster. Got it. Who else? You're taking it right now? Did you get it? Anybody else? Did you not get it? What's your name? Juan Garcia. Juan Garcia. Got it. Who else? You didn't do it?
I have my laptop.
What's your name? All right, I got you. Yep, app isn't working. Web browsers seem to work. Yep. Desiree Reyes. We will get you on here. Yep, Desiree, we've got you. You've already hit it. All right, have a good one. All right, Andrea and Ambria, got you both. All right. So, all right.